What happens when a small piece of DNA can lead to big discoveries? In the heart of Watertown, Massachusetts, a nonprofit called Addgene is helping scientists around the world do just that, one plasmid at a time. We spoke with scientists and staff to learn how this unique organization powers breakthroughs in research and what it means for the next generation of scientific minds.
My name is Megan Rigo. I'm the Director of Antibodies at Addgene, and I've been here for 9 years. In my role, essentially, one of our big focuses this year is to try to make our own tools. So historically, Addgene has distributed other people's tools, which is fantastic, but we know that there's some holes in the catalog. So what we're trying to do is look through, find what those holes might be, and make tools that are useful to scientists. So a big goal this year is to try to make some of those tools and make them available to scientists.
So give us an example of what tools means in the scientific world.
Sure, so I'm specifically talking about DNA tools. So DNA sequences that are not available right now, we can go and put them on a plasmid. So they might help a scientist make a viral vector or make an antibody or produce a protein in E. coli, something like that.
And the need is huge, correct?
Yeah, absolutely. Pretty much any area of basic research when we talk about the life sciences begins with a plasmid, because plasmids can make proteins, and proteins are so important. They can be enzymes, they can be antibodies, they can be therapeutics. So really, whether you're talking about pharma or you're talking about a small biotech company or you're talking about an academic scientist, all these labs are all using plasmids.
And before Addgene existed, what did scientists or companies that needed what you're describing do?
So if you needed a plasmid before Addgene, you would have to email the person that made the plasmid and ask them for it. And most scientists are really great about sharing, but if they get a message in their inbox asking for a plasmid, they might forget about it, or the lab might if you're looking at a paper that was 10 years ago, that lab might not even exist anymore if the person retired. Sometimes labs don't keep great track of their freezers, so they might send you something that isn't even what you needed. They made a mistake. So, before Addgene, there was a process to get it. It was not perfect. And there's also needs to be legal agreements between institutions, because plasmids, they're owned by the institution that made them. And a lot of people don't want to do the legal legwork. So Addgene also helps people through those legal agreements and makes it easy for them to get the stuff that they need.
Are there other companies doing what you're doing?
Really not Addgene, and not in a mission-aligned way. We're a nonprofit. We're not trying to, make millions of dollars here. We're really trying to help scientists, and I don't think you see that anywhere else.
Do you always have what people need?
No, we don't always have what people need, and I think that's why this year one of our big focuses is to try to figure out what we don't have and proactively make those for scientists.
I'm Hannah, and I'm a member of Addgene's scientific support team. So basically, my team helps our customers and any scientist that wants to send us materials basically answer any questions that they might have about either materials they received from us or sending us those materials.
So if someone comes to you
Yes.
and says they want something that you've never heard of before, what would you do?
Yeah, we get that question a lot. Addgene has like probably, I think we have over 100,000 plasmids in the repository. There's no way any one person could memorize them all. So we have scientists come to us and ask us for plant plasmids, mammalian plasmids, viruses. So basically for that, I start by searching our database to see if we have anything related. And sometimes this takes us into going into the scientific literature, kind of seeing what they're doing, and hopefully getting an answer back to them that is related to what they want.
And is it as easy as if you go to a store where there's a great demand for something, they start stocking more and more of it? Do you literally do that here?
Yeah, currently we don't make custom reagents. Basically, scientific labs will send us materials that they're working on. It does mean our materials end up kind of following the trends. For example, like when COVID happened, we had companies and labs suddenly, they wanted to send us all their COVID plasmids so we could help distribute those to scientists studying COVID. We're not necessarily custom making things, but what we have does follow kind of the trends in the scientific community.
Hi, I'm Naila Naeem. I'm the Scientific Support Manager at Addgene.
Tell us, what does a scientific support manager do?
Well, we get to help scientists across the world. We're basically like glorified technical support where we help them resolve issues with their samples and just answer questions. Sometimes we help them find materials at Addgene. Sometimes we help direct them to other resources and provide educational resources.
It's pretty heady stuff around the world.
Yeah, at least over 100 different countries.
So you've been here 5 years, is that correct? And
Yeah.
tell me, in that, prior to that, what were the 1 or 2 things that you did in the scientific world that led you to here?
Yeah, so I originally took an academic route. I got a PhD in Pittsburgh. I did a postdoc in Vermont, and I really like to study how cells communicate. So I was studying how a single molecule can, inform a cell what to do based on where it is. I studied how cyclic AMP stimulates proliferation in thyroid cells. But what that means is I looked at how a single molecule can tell a cell what to do depending on where it is in a cell. This involved using optogenetics, which are light-sensitive proteins. So I spent a lot of time shining a little light on cells to see how and learn more about how they grow. So I ended up at Addgene because I realized I wanted to do something a little bit more than just experiments at the bench. I wanted to help the broader community. So I was looking at other nonprofits to see what I could do, and found Addgene. I really enjoyed their mission about promoting and accelerating science, sharing materials, and I thought my expertise in biochemistry would be useful here. Since I've been here, I've really loved it. I feel like I've learned so much more here, working with all the different materials and talking with scientists around the world, and just getting to literally help people every day.
Given what you guys do you ever feel like you're biting off more than you can chew? I mean, because when you think about the scientific world, there's so many different aspects to it, and you're expected essentially to know it all in some form or fashion, right?
Yeah, I actually think that's one of the coolest parts because we have a very broad catalog of stuff. For example, I work a lot with DNA. Even though we have DNA that is for studies in mammalian cells and worms and frogs and fruit flies, just knowing some of the basic fundamentals about how DNA works, I can apply that to some of the different questions we're answering. So it's actually more like a fun challenge.
Talk to me a little bit about students who are thinking about becoming scientists someday. Is this the kind of thing that they would aspire to get into right away?
I think the earliest you can get into it, the better. And I think there's easy ways to do that. You can look at news articles about science. You can take out scientific books. You can buy little science kits, or I know some libraries too will have little microscope kits and stuff that you can take out. And I always tell people when they ask me what I do in the lab, I say it's a lot like baking. Cooking's funny because you can throw stuff in and it'll still taste great. But baking, if you don't follow the recipe, it comes out terribly. So I tell people if you're a good baker, you'll be a really great scientist.
And do you have advice to students who have a spark, who have an interest in science?
If the high school does offer AP courses in the scientific areas you're interested, AP Bio and Chem, those are great resources to start. But there's also other areas. The Boston area is awesome for bio and biotech and such. There's all sorts of universities, and many of them I think do summer programs or collaborations. So I suggest looking into those, see if maybe you can work in a research lab for a couple weeks over the summer, or some programs may have camps.
And so now I want you to close your eyes and I want you to think for a second back to when you were in middle school, and what was the thing that sparked you to get interested in science?
I was always really curious. I always had my hand up. So for anyone out there that's always got their hand up and maybe they're getting the side eye, keep your hand up. Follow your curiosity. I like science because I got to ask so many questions, and eventually I found the teachers couldn't answer my questions. So I had to learn how to find them myself. And by going through the scientific field, I learned how to do research, ask my own questions, and get answers. So that was why I ended up in science.
From DNA to antibodies, the team at Addgene isn't just moving materials, it's moving science forward. Next, we took a walk through Addgene's lab facilities to see how a plasmid goes from a freezer to a research lab half a world away. From biosafety checks to DNA sequencing, every step of the process is built on precision and care.
My name is Marcy, and I'd be glad to show you Addgene's lab.
Thank you.
So we're starting here in our bacteria labs. So scientists send us their DNA for us to store and distribute on their behalf. And once the DNA arrives here, we scan it into our system, and then our first step is to take the DNA and put it into bacteria for long-term storage and distribution. So we use our liquid handling robot to help us do these processes. Although it's not running right now, the robot can help us put the DNA into bacteria in a high throughput manner. So it really allows us to process many samples at the same time while allowing us to work in a very clean and contamination-free way.
And it's as simple as someone will call up and say, we have some DNA to drop off, and they'll fill out some forms and drop it off, and there you go.
Exactly. Yep, we have an online process. What they do is they submit their general plasmid information to us. They send us their address. We send them a kit for them to package their samples in, because we want to make sure everything is shipped according to proper shipping regulations for biological samples. So we send them all the shipping materials, the proper way bills, and then they ship it back to us, and everything is barcoded and scanned into our system when it arrives.
And I know this is a horrible question, but is there a vetting process at all, or any kind of?
The only vetting process we do is based on safety. So the lab we're in right now is rated for biosafety level 1 work, and the plasmids that we distribute are primarily distributed in lab E. coli, which is safe to work with at a biosafety level 1 lab environment. So that's the only thing we look for, is making sure that the plasmids are biosafety level 1 and they don't contain any toxins that we are not legally allowed to distribute for safety reasons.
And how often does that happen?
Every once in a while. We have a process where if something kind of flags in our system as potentially being unsafe, we will send a safety questionnaire to the scientist and say, what are the safety precautions you've taken with these? Can you work with them at biosafety level 1? And they send us their answers. We do need to reject some plasmids for safety, but most of the time, everything can be sent in to us for processing. All right, we want to head this way. So after the plasmids are put into bacteria, we want to do a quality check on them as part of our, storage and quality process before we distribute them to other scientists. So interestingly, after the plasmids are put into bacteria, we take them back out of the bacteria in order to make sure that what we put into the bacteria was correct. So when the plasmids are in the bacteria, we grow them up in large batches. We take part of that and we use that to make our freezer stocks. And our freezer stocks are what we use to distribute plasmids to the community. The other portion of that, we pull the DNA out and we sequence the DNA sequence. And we do that using next generation sequencing technology. And that allows us to verify that the plasmid that we have now stored in our freezer is going to be the exact thing that we were sent and the exact thing the scientist expects to receive from us. After our plasmids have been quality controlled, a subset of them can be used to feed into some of our other more specialized services. So beyond just distributing plasmids, we also distribute ready to use AAV preparations, which is a type of viral vector, as well as recombinant antibodies. So in order to support those services, we take the plasmids that we have in our repository and we make large-scale batches of them to then put into production for these other materials.
So dumb it down for us.
Yeah.
Plasmids are?
Yep, so plasmids are small circles of DNA. It contains information that scientists can use in their research. So they're basically genetic tools.
And it's the most common tool known to scientists.
Absolutely, and like the molecular biology, like almost everybody uses plasmids at some point in their basic science research in there. So oftentimes plasmids can be used just as they are as part of the research. Sometimes you have to use the plasmid to make something else. So that's what our specialty services do, is we take that extra step that a scientist would need to do in their lab and we do that for them. And that way we can ensure that they receive the same high quality, reproducible type of material that they come to expect with our plasmids, but now we can apply that to our viral vectors and our recombinant antibodies as well. So we have over 150,000 plasmids in our repository, and that's growing every week. We add about, I would say, about 1,200 plasmids each month to the collection with that. As far as a scientist coming to us and say, I can't find exactly what we want, do you have this? We don't offer those very customized services at this time, but oftentimes we can search through our vast collection and find something that's pretty darn close and often good enough for what they need. So after we have the large amounts of DNA to feed into our services, we need to move to a different lab environment. So our viral vectors and our recombinant antibodies require a higher biosafety level because of the nature of the way we produce them. So if we come this way, through here, we're not going to enter this room over here, but this is our biosafety level 2 suite. This is where all of our viral vector production and our recombinant antibody production takes place. So it's something that requires keycard access, additional training, and we have higher safety precautions in place. It's also noted that when you're working back here, you're wearing a blue lab coat, so we have the visual cue of I'm in the wrong place if I'm in a white lab coat and I'm on the other side of this door. The last kind of stage of a plasmid or a viral vector's life cycle is the getting it out to a customer. So we do all of this work to have these banks of plasmids and viral vectors and other ready-made products. But the whole goal of Addgene is to make sure these materials are accessible to the scientific community worldwide. So the last step of the process is order fulfillment. And this starts right here with the plasmids. So we have all 150,000 plasmids in these freezers. And every morning we have an excellent set of lab technicians who come in and they pick whatever plasmid somebody has ordered and they make a growing stab vial, and that will eventually go out to the customer the next day. So they pick roughly 600 stabs each day for shipment. So because they are in bacteria, they need to grow overnight. We want to make sure we do see good growth before we send a stab culture out to a customer. So they will pick it, they'll let it grow overnight, and then they hand it off to be packaged and shipped out to the customers. The last step here is our shipping room, and this is where all of our room temperature and temperature sensitive materials can get shipped out. So similar to the plasmids, all of our viral vector and recombinant antibody samples are in these banks of freezers here. So we have roughly 1,000 different viral vectors available and a little over 200 recombinant antibodies. And the viral vector samples are temperature sensitive, so they must stay frozen at all times, and we ship them out on dry ice. Plasmids, on the other hand, we can ship at room temperature, and they have, a little bit less coddling that is required to make sure that they get to a scientist in correct working order. So from here, this would be where our dry ice shipment and packing takes place. All of these blue bins are filled with dry ice. So we have large shipping containers that we fill with dry ice. We put the materials in, and then we make sure everything is packaged according to proper regulations for shipping internationally with dry ice or domestically.
On the list of things that are most important for you to be able to provide this service, what are the 2 or 3 most important things?
That's a very tough question to answer because there are so many different things that Addgene can provide. There are the physical samples, which we've talked about a lot in the lab, but then there is the additional support kind of behind the scenes that we provide that is equally important. So we have a very wonderful staff of scientists who provide technical support if scientists have questions about the materials. We have a great team of individuals doing all of the legal paperwork that is required to transfer materials between scientists. We have a great set of distribution workers who make sure we can clear customs in different countries with that. And of course, we have our in-house product and app dev team who are sitting, kind of in the center, building the software we use to be able to take in orders, to track all of our samples and make sure the orders get out to the right person at the right time. So really, I think it's hard to distill down to 1 to 2 things because it really is this interconnecting web of individuals and services.
I wonder if you could just quickly talk to the students out there who are science-centric and thinking about making a future out of science. As you said, you got your PhD and you came right here and you've been here for 14 years. Yes. So really impressive. Tell me why a scientist would be just as happy here in all the different things that you talked about doing as opposed to going into research or going into teach or going into another area.
So the great thing about Addgene is you can use science in so many different ways. You can be a traditional lab scientist at Addgene. You can be a scientist who doesn't walk in the lab at all and you're just helping other scientists do their work better. You're still thinking about science, but you're not doing the science. You can leverage your science into a business type situation where you're talking with scientists and you need to speak that language, but you're not actually doing science anymore. So I think that there are so many opportunities all under one roof at Addgene with that. But even just the basic science education, we have scientists at all different spans in their career. We have people who have taken maybe one science class in school who come in and work and are successful. We have people who have been CSOs at different companies and they come to Addgene and are successful in their roles. And everybody can contribute equally to our mission.
You seem unique. Addgene seems unique. Why?
So part of what makes Addgene unique is that we were first in a lot of the plasmid sharing plasmid repository. We were kind of the pioneers in that idea. So being first gives us a lot of room in there. There are definitely other repositories and other cores doing similar things, but many of them are located in different places throughout the entire world, throughout the country. They might be having a for-profit model where we have a nonprofit model, and that gives us a really great way to partner with the community of scientists we work with. We can come in and say, we're a nonprofit, we're mission driven, our purpose is to help you do your science
They
better.
know they're getting genuine service.
And they know it's genuine. They know, since we've been around for 20 years, that we are trusted. Our products have, had a lot of good word of mouth. We have good, high quality products, and people know that, and they now think of Addgene when they think of plasmids and viral vectors and recombinant antibodies.
Thank you.
Yeah, thank you.
But it's not just the labs that makes Addgene run. From shipping to software, from operations to outreach, it takes a whole team to keep the engine of scientific sharing going. Director of Operations Donnie Donaldson gave us a tour of their workspace in Watertown. So we are here with Andrew Donaldson, better known as Donnie. And Donnie, your position with Addgene.
Yeah, I'm the Director of Operations here at Addgene. I oversee our shipping operations as well as our facility operations. This is the Lynx building. I'm here in Watertown, Massachusetts, really close to the Arsenal Yards shopping district. Lots of food, gyms, Target's also just over that way. So this space we moved into 2018, fall of 2018, and so we've been here for almost coming on 7 years this coming fall. We also have two subtenants on this first floor. So this whole south first floor, so facing this way, is under Addgene, the Addgene lease. There's still 7 tenants, I believe, in this facility. Used to be an old Verizon warehouse, and so it was refitted to support the biotech industry. And that's kind of Watertown's growing into that new kind of biotech hub, the new Cambridge. Really happy to be here, and it's a lot of amenities. So we have our cafe, there's a bike room, showers for anyone who goes to the gym, works out, runs into work, bikes into work. And so a lot of great amenities for us.
Keep everyone happy and healthy.
Keep everyone happy and healthy. And then the outdoor space is really, it will get there because it's New England spring right now. So everything probably in the next 3 weeks, it'll just be a really nice spot out here. And the community of Watertown likes to walk through here and enjoy the park. And it's, it's outdoor space. And also it's a nice thoroughfare along to go to Target and the Arsenal Yards for shopping. So this space in here actually used to be a larger office space. During COVID, we actually reclaimed it, or the lab reclaimed it for lab space as we built out our antibody service. And so it used to be a little bit more open that way, and so we brought this in. So that was one thing during COVID that we took advantage of, the flexibility of square footage and less need for physical office space. And focusing on our lab needs. And we are in, our space here is between the lab and the office space is right around 30,000 square footage. About 60% of it lab, and say 55% lab, 45% office space. So you could almost say 50,000 square footage for each.
And I'm curious, as you bring up about office space and the pandemic and post-pandemic, is there a work from home option?
We do, yeah, depending on the team and the function. We do have many Addgenes, as we call them, individuals here that that are on this hybrid schedule. We have some that work from home permanently. It really kind of depends on what their needs are. And that's one thing that we, as the facilities team, have been really trying to support is that hybrid function. As you'll see, and even right over here, we have our hoteling space. So if anyone came in, we have a setup for them to pop down, plug their computer in, and then they can close this down. I mean, I've taken the space. And that way, we really try to facilitate those that have that, the hybrid work needs, so they can come in and be productive when they come in, depending on whatever space they need. So we spend a lot of time in this room. We also have social hours, which we're gonna have one later today, actually. I think they're actually setting up for it. I think we're playing Mario, one of the Smash Brothers tournaments. We did a Mario Kart tournament a few months ago, but this is really where a lot of people congregate. Social, coffee, all that stuff, and gathering around this table is a lot of fun times. We also name our conference rooms or our multi-use rooms after plasmids that are popular in our repository. And we actually voted, and this was before we moved here from the Cambridge office, we voted on what those names would be. And so we have Sleeping Beauty here, Groucho here. One of the big conference rooms over there is Sonic the Hedgehog, so we call that one Sonic. So it makes it kind of fun. We have guests when we're like, oh, meet me in Sonic. We're like, what are we talking about? So this is our room for it's our multi-use, but I would say like wellness room with puzzle room, nice place to get away from the desk, get some nice natural light. We have a lot of puzzlers here, so they move through puzzles like nobody's business.
How many employees?
Right around 120 employees. Fluctuates up and down. When I started here almost 7 years ago, we were closer to 80 to 90 employees, so we've grown since then. And we're about, when it comes to our hybrid work environment, we probably have about around 50 or 60 in the office on a daily basis. It fluctuates depending on, like today, a lot more because of our on-site meeting, our company meeting, so it fluctuates, but
and for aspiring scientists, this is obviously a place that would be of interest because it's not just one type of job here, right?
Exactly. There's so many different ways. We have our scientist team that can help work with other researchers that are looking for utilizing our material and helping them understand what they can use it for, helping them troubleshoot. So there's a lot of science being done that's not necessarily in the lab. And so in a lot of our researching new products and new services for our customers. So listening to our the scientific community of what they're looking for, what's new out there, what's new and novel that we can help them, as a repository, redistribute to the rest of the world. So as you've probably been hearing, is like a lot of what Addgene is helpful to do is let scientists do their science. We help, get their research material to everyone that's able to use it, so they can focus on their science and not the logistics and distribution side of things. This is my face on the wall. Yeah, one of my friends sent me this as kind of like a gag gift or whatever, and when I was moving offices, we found it, and then it's just been hanging around. So we've just kind of, and it's because I used to be back here, and so I think for the group over here on the viral vector team I think they're happy to see my face when they look up that way. So because they miss me, right?
I don't see any dart marks.
No, not yet, not yet.
At Addgene, it's not just about the science, it's about sharing it. Whether you're in a classroom or a lab, in Watertown or across the world, the team here is proof that open access can drive innovation. For the next generation of scientists, the tools aren't just in the lab, they're within reach.